Alright, class, so we're going to start talking about biological molecules. Alright, so before we start talking about specific groups of biological molecules, alright, the first thing we have to talk about is how all these molecules are assembled and disassembled. Alright, so when you're talking about biological molecules, you're talking about macromolecules. Alright, so really large biological molecules. Alright, but these large biological molecules are actually made up of individual building blocks. Alright, so think about a house that's made out of bricks. Alright, that house isn't made out of one large brick, right? It's made out of individual bricks that are stacked on top of one another. Okay, so the same idea applies to biological molecules. Alright, so these macromolecules, a majority of them we refer to as polymers. Alright, so they consist of a series of similar building blocks that are covalently bonded to one another. Alright, so these biological molecules that we consider polymers are your carbohydrates, which we'll talk about just a little bit, your proteins, and your nucleic acids. Alright, so now there are 4 classes of biological molecules. Note that I've only mentioned 3 of them. Alright, so the only group of biological molecules that we don't consider polymers, alright, are going to be your lipids. Alright? So your lipids do not form polymers, or they're not made of polymers, okay? Or they aren't polymers, okay? So talk about a polymer. It doesn't matter whether I'm talking about carbohydrates. It doesn't matter whether I'm talking about proteins. It doesn't matter whether I'm talking about nucleic acids. They're polymers. And polymers are assembled by their individual building blocks that we call monomers. Alright, so I've kind of keyed this image and color coded it. Alright, so here in black. Alright, this is simulating the polymer. Alright, so this is the polymer. This entire chain of links. Alright, that represents the polymer. Now, each individual link in red is what we refer to as a monomer. Alright, notice that they are similar things. Alright, so in this case we're talking about links in a chain. Alright, so as we move on into each class of biological molecules, each class has its own building materials. Alright, so you use bricks to make a brick house. Alright, you use wood, different types of wood, to make a wood house. Alright, so each of these molecules requires different building blocks. Alright, but they're all going to be covalently bonded to one another. Alright, to form their polymer. Alright, so this is kind of a short overview of polymers.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Alright, so they talked about 2 different types of reactions. Alright, so you have dehydration reactions and you have hydrolysis reactions. Alright, so dehydration reactions are used for synthesis. Okay? So you're making a polymer. Alright, so here you're given an example of where you have 1, 2, 3 monomers that are linked together. Alright, so they form a short polymer. And here, at the very end of your polymer, you have this little bitty hydrogen atom that is sticking out. And here you have another monomer that's at this point in time it's free. It's not attached to anything. Okay, but it has a free OH group. Now, when you are synthesizing polymers, you use a dehydration reaction. Alright, so what happens when you dehydrate? You are losing water. Alright, so what's going to happen here is this polymer and this individual monomer, you're going to have the removal or the dehydration of a molecule of water. Alright, and so that's what's taking place. This water molecule gets plucked out, and you have the formation of a new covalent bond. Alright, and your polymer has now increased in length by 1. Alright, and this is a continual process. Alright, so you want to add another monomer, or it involves another dehydration reaction. And it can get longer and longer and longer. Alright, it doesn't matter, again, whether I'm talking about carbohydrates, whether I'm talking about your proteins, whether I'm talking about nucleic acids. When you're synthesizing the polymer, it involves a dehydration reaction. Now, looking at the opposite, alright, so in this case, talking about degradation. Alright, so when you are breaking down, tearing down a polymer into its individual monomers, you start plucking off monomers. Alright, this involves a hydrolysis reaction. Alright, so a hydrolysis reaction or hydrolysis reaction. Alright, think about that word, hydro, water, lysis. Alright, it involves the use of water, a molecule of water, to come in and break the covalent bond. Alright, so here you're breaking that covalent bond by adding a water molecule. And when you do this, you release one of the monomers. Alright, so if you wanted to break this other bond, it would involve another water molecule. If you wanted to break this one, it would involve another water molecule. Alright, so each time you break off an individual monomer from its polymer, it involves a water molecule breaking that covalent bond. Alright, so dehydration reaction, synthesis. Degradation, hydrolysis reactions. Alright, you need to be able to keep this straight, all right, because this is important when we go through each of these other biological molecules, because we're going to talk about how they are assembled. And we're going to talk about the individual building blocks. Alright, so what this is illustrating is basically you're looking at the electrons that are being shared between atoms. Now, what you're looking at here is, in this case, look at where the electrons are located. Alright, so if they're located in the middle, alright, you know that you're dealing with something like a non-polar covalent bond. Okay? But then when you're dealing with something like here or here, where your electrons are more closely associated with one atom in that molecule, alright, you're dealing with basically a polar covalent bond. Okay, so why am I bringing up or tying back in this idea of chemical bonds and electronegativities? All right, because that's what you're dealing with here. The oxygen atom is more electronegative than carbon. All right, well, the reason I'm bringing this up is because it has to do with the amount of what we call potential energy that's actually stored in these molecules. Alright, so carbon dioxide, there really isn't a lot of potential energy stored in that molecule. Very little. Alright. Your carbohydrates, alright, you have a lot of carbon-carbon, carbon-hydrogen bonds. Alright. Basically, there is a large amount of potential energy stored in these bonds. And when you break those bonds, guess what? You release energy. Now, when you look at a fat is a hydrocarbon, right, which we'll get to a little later. All right, but notice here that chock full of potential energy. All right, so fatty acids have a higher amount of potential energy than carbohydrates. All right, both carbohydrates and fatty acids are a source of energy, all right, for our bodies. As we break down these molecules, all right, they're a good source of fuel, right? All right, these basically lead to the production of ATP. But guess what? You get more ATP from fatty acids than you do carbohydrates. All right, so think about chemical reactions. All right, so talking about the amount of potential energy. All right, so the amount of potential energy stored in a molecule. All right, so there's more potential energy stored in the hydrogen gas and oxygen gas molecules themselves. All right, these electrons are held very loosely. All right, so they're very easy to kind of pluck off and pull away. All right, but when these atoms get rearranged into molecules of water, all right, there's a decrease in the amount of potential energy. There's energy that's lost, right? Because you're breaking and forming new bonds, energy is released. All right, these electrons that are associated with the oxygen atom are held much more tightly to the oxygen atom than the hydrogens, because oxygen is more electronegative. All right, so there's a lower amount of potential energy there. Alright, so going back here, note, fatty acids, tons and tons and tons of nonpolar covalent bonds. Alright, so basically when these nonpolar covalent bonds are broken, guess what? You are releasing all of that stored potential energy. And guess what? All that stored potential energy comes back to form ATP. Alright, so here, alright, talking about that process. Alright, so for example, you have sugar, glucose. Alright, so glucose is a sugar, is a type of carbohydrate. This molecule is highly ordered. Highly ordered. There's a higher amount of potential energy stored in this molecule of sugar. Alright, so go back here. Look at your carbohydrates. A high amount of potential energy. Alright, so here you have 1, 2, 3, 4, 5, 6 carbon atoms. Alright. Alright, so another sugar, basically. Something similar to glucose. Alright, so highly ordered structure, high amount of potential energy. Alright, you add oxygen. You basically go through a process of a combustion reaction. Alright, and you release heat. You also end up producing ATP. But note that your products, your water, and your CO2 have a lower amount of potential energy than your initial reactants. Alright, so there's an exchange, a release of energy. That energy is captured, used by the cell to make ATP, and then a good bit of it is actually lost in the form of heat. Alright, so moving on into carbohydrates. Alright, so your carbohydrates. Alright, so carbohydrates are classified based on different sets of criteria. Alright, now your carbohydrates are going to contain carbonyl groups. Alright, so recall what a carbonyl group is. Alright, so a carbon atom double bonded to a oxygen. Alright, so that's a carbonyl group. You have hydroxyl groups, which are OH groups, and then carbon-hydrogen bonds. Now, carbohydrates, there's a chemical formula for carbohydrates. Alright, so you'll want to kind of commit this little formula to memory. Alright, so CH2O and then N. Alright, so here N has to be 3 or greater. Alright, so your simplest carbohydrate would be something like C3H6O3. Okay, that'd be one of your simpler carbohydrates. Now, something you guys are probably more familiar with is something where N, instead of equaling 3, N equals 6. Alright, so if N equals 6, guess what? You get C6H12O6. And guess what? That's our good trusty friend glucose, for example. Now, monosaccharides. Alright. Monosaccharides is the term, the label, that's used to describe the building block of all of your carbohydrates. Okay, so your monosaccharides are single sugars. monosaccharides are the monomers, the building blocks of your larger carbohydrates. Alright, so monosaccharide monomers, they can vary in different ways. Alright, so location of a carbonyl group, we talked about this in the last chapter. Alright, we talked about aldoses and ketoses. Alright, we talked about that you're in an organic molecule, you can have a variety of carbon atoms that vary in length and number. Alright, so your simplest carbon or organic molecules, your carbohydrates, would have 3 carbons, so triose. Alright, 5 carbons, pentose, 6 carbons, hexose, 7 carbons, for instance, would be a heptose. Now, these monosaccharides, there's a spatial arrangement to their atoms, and you can have different arrangements of hydroxyl groups, all right, which we'll get to. And then your monosaccharides can exist in a linear as well as a ringed form. All right, so we talked about linear formed and ring formed or cyclic form of organic molecules. All right, so here, looking at your aldose and ketose. All right, so recall that your aldoses consist of a carbonyl group at the end of the molecule. Your ketose consists of the carbonyl group within the backbone structure. All right, so again, glucose and galactose. All right, they have the same chemical formula. All right, but guess what? They are going to be isomers of one another. Now, looking at glucose and galactose, they're isomers of one another, but why exactly are they isomers? Now, when you look at glucose and galactose, all right, they are both what we call aldose sugars. All right. Now, what's the difference between glucose and galactose? Well, note how it previously mentioned that the spatial arrangement of your hydroxyl groups matters. Well, here, basically, you have your OH group on this 4th carbon here. It's basically pointing up. All right. And then you have on galactose, you have the same carbon and OH group, but it's pointing down below the plane. All right, so in glucose, it's pointing above the plane. In galactose, it's pointing below the plane. All right, so the spatial arrangement of these chemical groups varies. All right, so they're isomers of one another. But it's not because of whether they're aldose or ketose sugars, it's because of the spatial arrangement of their OH groups. Alright, so this kind of gives you a breakdown of the triose, pentose, hexose sugars. Alright. And it gives you an example of an aldose and a ketose sugar for each. Alright, and it shows you where that particular functional group is located. Alright, for each. Now, ribose. Now ribose is interesting because ribose is a pentose sugar. Now ribose is a pentose sugar, okay, is a component of your nucleic acids, okay, which we'll get to when we talk about nucleic acids. All right. Now, your hexose sugars. Now, if you compare glucose and fructose, glucose and fructose look exactly the same in the sense that their chemical formula is the same, but structurally they're different because they're isomers, but they are structural isomers. All right, because this has a aldose, this has a ketose. Now, when you compare glucose and galactose, they're both aldoses, but the spatial arrangement of their OH group differs. Then when you compare galactose and fructose, all right, you're comparing a aldose and ketose sugar, but you're also comparing an alteration in the OH group as well. Now, this is showing you how these linear forms of your carbohydrates cyclize. Alright, so that's what's taking place here. Alright, so you have this chemical group here at the end of the molecule. You have this OH group here. Basically have the formation of a covalent bond between these groups to create your ring form. Alright, so when you're talking about sugars in an aqueous solution, now remember, an aqueous solution means that the solvent is water. Alright, so in presence of water, your linear form of your carbohydrates will cyclize. They'll form the ring structure. And this will be the more predominant structure in an aqueous solution. Now, on a bench top, all right, in a sealed container, most of the sugars are going to exist in the linear form. Now, what this is showing you is the fact that you can have two different types of or versions of the same sugar. All right, so for instance, you can have glucose. You can have alpha and you can have beta. All right, so depending on whether this OH group goes basically below the plane of the molecule, all right, so create a plane line. All right, so if this OH group is below the plane, it's an alpha glucose. All right, because this sugar is glucose, this OH group is below the plane, so it's labeled as alpha. Your beta glucose, it's above the plane. All right, so it is a beta glucose. It's a glucose molecule, but this OH group is above the plane. All right, now alpha and beta glucose monomers are going to be important when we start talking about your polysaccharides.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Alright, so disaccharides. Disaccharides consist of two monosaccharides, two single sugars, that are joined together. Alright, so they're joined together by a covalent bond. Alright, and the name of this covalent bond is what we refer to as a glycosidic linkage. Now, you will want to commit this to memory. The covalent bond that links or joins two monosaccharides together is a glycosidic linkage. Now, again, since you are forming, you're elongating, you're making a larger molecule. Alright, this is a synthesis. Alright? So since you're synthesizing something, you're making it bigger, making more of it, okay? This involves a dehydration reaction. Alright, so you have the removal of a molecule of water. And you have the formation of your glycosidic bond. Now, maltose. Maltose is composed of two monomers of glucose. Alright, so maltose is malt sugar. Alright, the malt flavoring you find in your favorite either chocolate or vanilla or strawberry malts. Alright, they use malt sugar. Maltose is a disaccharide. Now, something like lactose. Lactose is composed of glucose and galactose. Alright, so glucose is a monosaccharide, galactose is a monosaccharide. You join these two together, you get lactose. Lactose is the sugar found in milk. It's milk sugar.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay, so looking at your polysaccharides. Alright, your polysaccharides are the polymers of your carbohydrates. Alright, so your polysaccharides have two major functions. They either function as a storage of fuel, energy, alright, or they play some structural function, alright, for the cell or the organism. Now, again, I'm going to tie in this idea of structure and function. Okay, so the molecular structure, the arrangement, the order of these particular monosaccharides, the specific type of monosaccharide, how they're strung together, is tied in with the overall function of that polysaccharide. Alright, so we're going to kind of go through this list of polysaccharides over the next couple of slides. Alright, so this kind of gives you a kind of a view of each of these types. Alright, we're going to kind of walk through each of these. Alright, but you can already note some of the differences between starch and glycogen and cellulose and how they look. Okay? So, before we start talking about all these, it's important to kind of talk about the chemistry behind it. Alright, so you have alpha and you have beta glucose. So again, alpha glucose, the OH group is below the plane. Beta glucose above the plane. Alright, so starch. Starch is made up of alpha glucose monomers. Alright, so that means that it consists of individual alpha glucose sugar molecules covalently bonded to one another. Now, cellulose consists of beta glucose monomers. Now, this is important, alright, because here you'll notice that the OH group alternates. The sugar molecule flips. It goes from being the OH groups on the bottom to on the top, on the bottom to the top. So the sugar basically, since this is a single bond, all right, we talked about how single bonds, they can rotate. All right, that's what's taking place. The sugar molecule is rotating on its axis. All right, so you have the OH group that's alternating. All right, so beta glucose monomers alternate. Now this is important. All right, we'll get to that, get to why a little bit later, but it is very important as to why cellulose is arranged the way it is and why starch is arranged the way it is. All right, because here you see all of these OH groups on one end, one side of your growing polymer. All right, so looking at starch. All right, so starch is the storage form of energy in plants. All right, so you basically have two forms: amylose, amylopectin. Amylose consists of a straight chain, which is the majority of it. Right, so majority of starch is amylose. All right, there's a smaller percentage of starch that's amylopectin, which is actually branched. It has branch chains coming off of it. And basically, when you think about starch, it can coil up. All right, so going back and looking at starch, all right, the structure coils on itself. All right, so this is only a small view of a molecule of starch. All right, but it can basically coil up in on itself like an accordion. Glycogen is the storage polysaccharide found in animal cells. All right, so it's very similar to starch. All right, but in animal cells, you have glycogen. All right, so glycogen is an energy reserve. So you find it in the liver and in the muscle cells. All right, so think about why your muscle cells would like to have this excess storage of glycogen. Well, your muscle cells are constantly contracting, and that contraction requires energy. It requires ATP. Now, where do we get ATP? Well, fuel, food, things like glucose. Well, glycogen is nothing more than alpha glucose, right? So basically, you can chop off individual glucose monomers and use that as a fuel source. You can convert your glycogen into glucose. Now, looking at cellulose. Alright, so cellulose consists of your beta glucose monomers. Now, again, look, you have alternating OH groups because the actual sugar molecule itself rotates on its axis. Now, this orientation is really cool. Alright, because look, you have a chain of beta glucose sugar molecules there. You have a chain of beta glucose sugar molecules there. You have a chain here and a chain here. Now, it's all held together. One chain is held to the next chain through hydrogen bonding. Okay? So basically the OH group on one of these sugars here, one of these glucose molecules here, can form a hydrogen bond with this one, and so on. Alright, so notice that the cellulose molecules are stacked on top of one another. This arrangement, this orientation, the flipping of the sugar molecules allows for these OH groups to interact with the chain of glucose molecules above it and below it, and you can form hydrogen bonds. Alright, so it's basically like laying sheets on top of one another. Alright, so cellulose. Cellulose plays a structural role for the cell. Alright, it's a component of the cell wall of plants. Alright, so this forms a nice barrier. It lays on top of itself. Alright, you have one layer here, and you have another stack. And then you have another stack. And then you have another stack. And it can keep growing. Alright, but each stack is held together by hydrogen bonds. Alright, so this is giving you kind of a closer image at cellulose. Alright, so here you're looking at a leaf, and you're looking at just an individual cell of that leaf. Alright, and you see this kind of faintly gold, yellow stained portion that's exterior to the cell itself. Alright, that's what we call the cell wall. Alright, now the cell wall is mainly comprised of cellulose in plants. Now look here, if you look at a closer image, notice that they basically consist of strips of cellulose fibers. Alright, so you have this long chain laid on top of one another. Alright, so it can basically, if you think about it, here, it can basically expand and contract. Expand and contract. Alright? It can stretch. I mean, think about it. It's cotton. Cotton, for this example. Cotton is stretchable. It's pliable. It has give. Alright? That's because of this right here. Its makeup. Its chemical makeup. The types of sugar molecules. The hydrogen bonding between each level or layer of cellulose chains. Alright? So if you only look at one microfibril and look at individual cellulose molecules, alright, so here, this is showing you the hydrogen bonding that's taking place between one chain and the next. Alright, so all of these consist of hydrogen bonds. Now, chitin. Chitin is another structural polysaccharide. Now, the structural polysaccharide is found in both fungi, is found in arthropods, insects. Alright, so for fungi, it forms the cell wall. It's a major component of the cell wall of fungi. Okay. Now, your insects and arthropods, it forms the exoskeleton of these organisms. Now, chitin here consists of what we call N-acetylglucosamine monomers. Alright, so you're not looking at glucose, but you're looking at another type of sugar. Alright, and this is a N-acetyl group. Alright, so N-acetylglucosamine, or very short reference, NAG. Now, this kind of gives you a overview of the different types of polysaccharides. Now, the one that I haven't mentioned, alright, is peptidoglycan. Now, it consists of N-acetyl glutamic acid and N-acetyl muramic acid. And these two sugar residues alternate. Alright, so you basically have NAG, NAM, NAG, NAM, and it forms this long chain. And these chains of sugar molecules make up the cell wall of your bacteria.